All right. So Leo works with me at the Quadram Institute, where he is the head of phylogenomics. He has been working there for 1 year, supporting and creating analysis of large data sets. Before that, he held postdoctoral positions at University of Lausanne in the Swiss Institute of Bioinformatics and at Imperial College London and at the University of Vigo in Spain. He mainly worked with the development of phylogenetic models and tools at the genomic scale, usually focused at the tree of life level. So, he did his PhD in the University of Tokyo with the K from HKY, right? Yeah. So take it away, Leo. Thank you so much.
All right, thanks. So yeah, I would like to start by thanking Nabil and Andrew for the opportunity of talking here. And so for this talk, I decided instead of talking about, let's say, new results, I'd like to do a presentation that reflects on my own path. So moving, as you said, moving from eukaryotes, viruses, and now to bacteria. And so basically, I would like to start talking about the trees of life, because I believe there are trees of life, not only because you might have a network, there's no single tree that represents, but also because at different resolutions, you might be interested in different things, and you might be seeing different aspects of this tree or of these trees. And so, if you start to zoom in into the tree of life, and then you may end up like this. So by the way, this is a recent work on the ribosomal RNA operon. And this shows that depending on which copy of the operon you choose, you end up having a different history. So if you take all of them together, you have this tree here. So, and then if you zoom in a different part of the tree of life. So for instance, now we're looking at fungi. And then we might have something like this. And so this actually, I'm sorry, I don't remember which gene family is this, because there were, I think, 12,000. But so this shows a gene family that we see duplications. So the two, can you see my pointer? Probably not. Okay, cool. So yeah.
No, I don't see it. No, I can see a pointer, yeah.
Yeah, you can see it moving here. So you have a blue subtree here, you have another blue subtree here. So you have the same species on the subtrees, because actually this gene family, it had a lot of duplications. And so what I want to say is that you have different, even in the same tree of life, you have different trees and different ways of looking at things in this tree. And so what's phylogenomics? So we know what's phylogenetics, right? You have your gene, and then you align this gene, and then you estimate a tree using your favorite method. However, if you start estimating different genes, different regions in the genome, you end up having different trees. And they are different not only because of stochastic error, not only because your method is not perfect, but it's because the true evolutionary history of those genes is different. So you have heterogeneity, you have paralogy here. So for instance, here we see two copies of, for species C, we have C1 and C2, D1 and D2. You also have a lot of missing data. And so for me, phylogenomics is when you take explicitly this true diversity into account, which, so this diversity comes because of biological processes like duplication and losses, or population processes like coalescences, or some people call incomplete linear sorting or ancestral polymorphism. And also, horizontal gene transfer, migration, hybridization, recombination. All of these are biological phenomena that make that the history of the species is not reflected exactly in the history of the genes. And this leads to new questions, right? So now we can start comparing these genes. So why are they different? Where are the differences? What's the reason for the differences? And then we can also do, I like this term gene shopping, because it's like a good version of selection bias. So you actually, now that you have so much data, you can choose the data that better follow the assumptions of your model. So for instance, if you want to estimate divergence times, you might want to look into genes that follow a molecular clock. And so that's what people are calling gene shopping. And also species tree inference. So now that we have the gene tree, what's the species tree? And I'm gonna focus more on this on the rest of the talk. Just a brief interlude, just to say that, so in statistics, you have two classes of problems, right? When you have too much data and when you have too little data. And in our case, so the samples are the sequences or the genomes, and what I call the features can be the genes or can be the columns in the alignment, which are the DNA bases or the amino acids. And then you might have a situation where you have a tall data, so you have a lot of samples but not many features. This is the problem of trying to estimate a tree of 10,000 species, but you only have one gene of 100 base pairs. And then you have also wide data, so when you have a lot of features but few samples, and here you end up having problems of collinearity, of noise. And so usually when we think about phylogenomic models, you have to think a little bit about which kind of, on which of these classes of problems we are ending up. And so during the rest of the talk, I'm gonna give an outline of this 5 classes of methods. This is a classification that it's very broad, but are the phylogenomic ways of estimating species trees from gene trees or from alignments. So classically, what's a phylogenomic species tree inference? Is something like this. If you have several gene families, now we have a lot of this kind of data. What can you do to estimate the species tree? Well, the most basic way, the most common one is called supermatrix. So a supermatrix approach is when you just concatenate. So first you align your gene families. And by the way, when I say gene family, I mean all the sequences that you assume have common ancestry, which in practice is all the sequences that you align and that you are pretty confident that you should be aligning them, not just aligning random sequences. So these are the gene families. At some point in the past, you assume that they had a common ancestor. And then if you can manage to concatenate these alignments, then you can use your favorite phylogenetic tree estimation software to find the species tree or the set of species trees. And then there's the alternative, which is for each one of your gene families, you estimate your gene tree. You apply, I don't know, IQ3 to each one of the genes, and then you have a set of gene trees. And then once you have this set of gene trees, and then you try to summarize the information from them into your species tree. However, in practice, usually they depend on the definition of the orthologous groups. So, for instance, we are familiar with Proca and Rory, and in Proca and Rory, you have to find the orthologous group such that you have, in the end, this alignment. And many methods, I think most of even the let's say the Tree of Life papers that we have, a lot of them still use supermatrix, so they still have to kind of find which are the orthologous groups. And here I'm showing a hybrid approach, which I'm classifying, I'm putting together in the same bag as supermatrix, which is what's called supergenes. So instead of just concatenating all your genes and estimating one species tree, or instead of estimating tree for each one of your genes and estimating the species tree, so this would be a super tree approach. First, you estimate the trees for each one of your genes, and then you look how incompatible are they? Because if the two trees are not completely incompatible, if they might have a common signal, and then you beam them into the same, what they call here a beam, and then these supergenes is the things that you can concatenate, and then you estimate the tree on them, and then in the end you apply a super tree approach. So it's a summary of your trees to have your species tree. So just to mention that I'm putting them in the same bag a super matrix. And so, in a nutshell, what are the advantages of this method? Well, they are easy to interpret because if you know phylogenetics, if you know how to interpret a tree, you know how to interpret a tree in this sense as well. And they are relatively fast, especially if you don't have too much uncertainty. It's relatively quick. You know, if you run IQ3, you know how quick it is. That's my measure of quick. But there's a problem that it neglects the phylogenomic heterogeneity. I'm not talking here about the rate, the site-wise rates, but I'm talking about what we mentioned before, if it has duplication or losses, or if it has a population structure. And so you lose this heterogeneity. And it relies on orthology assignment. So you rely on good databases that give you the orthologous groups or a good pipeline. So the next group of methods are what I'm calling here the centroid methods. So I'm putting in the same bag all the methods that try to minimize a distance between your gene trees and your species tree. So the most famous example here is Astral. So Astral is based on quartets. And the general idea is that you have your gene trees here, and then you calculate a distance. So it's a disagreement between each one of your gene trees and the species tree. And then you try to find the species tree that is centroid to all these gene trees. So it's closest to everybody. There's a gist here, which since we're talking about phylogenomics, we have to think at some point about paralogy or about populations. So for instance, in this case, G4, you have two elements from the same species. I have the color pink here, or color orange here. So you have two elements, yellow here. So this kind of trees, the technical name for them is multi-label trees, which are the gene families, because gene families, you have several genes from the same species. And then in this beautiful drawing, I'm representing how they solve this problem in the distance minimization context. So basically they say, well, your gene tree is multi-label. So it has several labels here, several tips that have the same label. Then you just need to do a trick on the species tree, and then you create an extended species tree. And once you have this, now they are comparable. Now we can use our favorite distance between trees method. So for instance, Robinson's fold. And by the way, this is called MURF because this is a multi-label tree, Robinson's fold distance. And then you can now calculate the Robinson's fold distance between your gene family and the species tree. And the newest version of Astral, I think it's using this or something very similar to this. The first version couldn't handle multi-label trees. So that's why I'm putting them in a different bag. And there's a closely related class of methods which are called reconciliation. So it's also a distance in a way, they call it a cost. And basically they say, well, if you have your gene tree and a species tree, how can you reconcile them? How can you put the gene tree inside the species tree? And there are very quick algorithms to find this under a population approach. So it can tell you the number of what they call the deep coalescences necessary to describe the difference between them. I'm sorry, I'm not going into the details, but it's a fast and easy to calculate. Not so easy, but it's doable. And also assuming duplication and loss process, you can use the same algorithm only with the twist at the end. You can also estimate the minimum number of duplication and losses necessary to explain the disagreement between them. So how can you reconcile these two trees? And under lateral gene transfer or recombinations, you can also use, this is not so easy to calculate, but you can also, at least in theory, think about how to describe this difference. So it's a reconciliation between the two trees. And again, you can use now these trees to find what's the species tree that optimizes this cost. These methods are fast. And actually, maybe they are fast because we're doing a trick here, right? So first you estimate the tree for each one of them, and then once you have the tree, everything goes fast. And it works with both tall and wide data sets. So they work with both data sets because of a trick that if you have a very long alignment and then you estimate the tree from this alignment, what you're doing actually is summarizing your features. So every feature now, so every column here now is not a site in your alignment, but actually is a tree. So even if you have, I don't know, 500 trees, 500 different genes, you are reducing quite a bit the size of your problem. But it's hard to interpret biologically because, this is basically, it's a mathematical construct. So it works well, but sometimes you feel short of an explanation of why the tree that you found is that one. And it also relies on you having good trees. So, garbage in, garbage out. If you estimate well your gene trees, and then this tends to give you good results under their assumptions. But otherwise, it can give you anything. And since it's hard to interpret biologically, you might be in trouble. And there's no, I should say, there's no parametric uncertainty. So it's not incorporated into the model how to describe the uncertainty there. Because, yeah, you can always do, bootstrap or some non-parametric way of finding confidence intervals. So this one I'm gonna talk briefly because I realize that it's quite complicated. I might get lost trying to explain this equation, so let's just skip it. No, but actually, so the multi-species coalescent process is an extension of the coalescent process. So coalescent process describes the evolution of populations. So when you have an explicit model for the effective population size and the diversity, and they can extend this model to when you have different species. So they say that, well, within a species, within a species branch here, you have a coalescent model, but then you can extend this coalescent model for when you have the speciation process. Because the definition, so this is one of the definitions of species, is when you start to exchange material between the populations. And so you can use this to write the probability of having a gene tree being generated by a species tree. That's going to be a product over the branches of under a coalescent model. So if you can't calculate this, and to calculate this, you need to calculate this, and this comes from the coalescent, that you see the term here of the effective population size. Anyway, you can calculate. So you have now we have a full probabilistic model to describe the coalescent process over different species. And this is famously the star-beast model. And we also have another Bayesian, another probabilistic way of describing this difference, which is Bayesian minimization. So I should say Bayesian regularization, because what you do instead of trying to find the species trees that minimizes the distance, you just say, well, if the gene tree is very different from the species tree, then the probability of the species tree generating this gene tree should be low. So basically, this is what this exponential distribution is saying. That the probability, so if you see a gene tree, the probability of this gene tree coming from that species tree depends on how similar they are. And once you define this, and since there are several ways of calculating distance, as we saw between species tree and gene tree, you can just add them into this equation. So this is the likelihood that IQ3, RexML calculate, and then you just add another parameter here, which says, well, now it's not enough to have a gene tree, but actually I want to know the distance between the gene tree and the species tree. And then if you put this into a Bayesian machinery, you can estimate the whole set of potential species trees and also of gene trees that are not in disagreement. And so these two methods, I bag them together because they're both Bayesian, they are both very slow, and they require some familiarity with the models, and so that's why I'm saying here that they are not user-friendly, but they can handle uncertainty pretty well, and they have an explicit biological interpretation. And so examples are StarBeast, FieldDog, and my own genome. I'm putting this, so there's another class of methods where we're reaching the end. There's only two more. So there's another class of methods which they are summary-based methods. They also call it patristic distance methods. They are really nice. They are really interesting. If you know, if you're a computer scientist, you'll love them because they're really fun to implement. Basically, they say, if you have a gene family tree, and then from this gene family tree, you can write down the pairwise distance matrix between the species. And then if you have a set of distance matrices and they are on the same species, you can summarize them. Let's say take the average, take the minimum or whatever. And in the end, you end up with one distance matrix for your species, which we all know and love. It's the neighbor joining or UPGMA. So you can apply clustering algorithms on this distance matrix now, and then you can have your species tree. There is just one problem, that there are a lot of combinations of ways of merging, of summarizing this information. So for instance, across the loci, do you take the minimum, you know, between these matrices here, do you take the minimum, do you take the average, the median, whatever? And when you have several individuals here from the same species, what do? Do you take the minimum, the average, again? And so that's why I came up with this. So this is like a toy software that you can play with different options for how to solve this in order to come with this tree. And then again, so you have several clustering algorithms, right, that can give you a dendrogram. And I like these methods. They are fun to implement. They are very, very fast. Because in the end, you know, you can reach, no matter how big is your data set, at some point you end up with one pairwise distance matrix that you just apply neighbor joining. So I think neighbor joining actually is the limiting factor here. It can work with very large gene families, and by very large gene families, I mean with a lot of duplications, or even if you just have 50 sequences, but then you have 5,000 representatives. So your gene tree has 5,000 tips. You can summarize it very quickly, and then you're done. But it relies not only on having good topologies, but also on good branch lengths. And this is a major drawback because the branch lengths, it's what's going to give you the distance between the species. And as I said, there are many methods to choose from, and you feel a bit lost reading the literature because there's no clear-cut best method. And basically, so that's why I came up with a software that implements all the combinations. And I think currently it has 72 combinations so you have potentially 72 trees using this class of methods. And in the end, I think one of my favorites at the moment, which are the alignment-free methods that are used for a lot of things, and amongst them, species tree inference, or let's say the tree inference in general. And thankfully, I don't have to give too many, let's say, detailed explanation about this because we are quite familiar with those. But just to mention, if you're not familiar, that alignment-free methods, they are based on the summary of your sequences. So once you have your sequence here, it calculates the k-mers, and then from the k-mers, it has what they call a sketch, and you have different kinds of sketches that you can have from your sequences.
Leo, I think your connection's a bit shaky.
Okay, do you want me to repeat this last part? Hello?
Oh,
Oh, okay. Yeah. So, so you have sketches, which are a summary of your sequences, and then you can use not only the presence or absence, but also the frequency of the sequences. And I think Nabil is on. Anyway, I'll keep on talking. And and then, so now that you have a sketch, which is basically something that you can, a good representation of your genomes, you can calculate a distance between them. And the assumption here, the phylogenomic assumption, is that the distance between the sketches can be used as a proxy for the evolutionary distance. And so in a nutshell, these methods, they are very fast. They have a good performance. It overcomes paralogy because it's not even an issue for these methods. However, this is a bit inflammatory, but there's no phylogenomic model for K-mer. So, if you want to use directly a K-mer model for phylogenomics, you have to remember that basically you are in the same line of reasoning as the cladists, saying that the distance between sequences is the distance, it's the evolutionary distance. And we know that in many cases this is not true. And due to that, you don't have, again, a parametric uncertainty for this. And then just to summarize, this is by popularity, by the way. I'm not saying that this is not a recipe. It's not what you should do, but just by seeing the literature and by seeing what my colleagues are doing, I think that when you work with strain level phylogenies, it's very common to use supermatrix approaches or alignment-free approaches. Then when you go to what they call shallow phylogenies, which basically are populations, so closely related species, then you start seeing the multi-species coalescent, summary-based methods. And then when you go deeper, when you start studying the tree of life, then you start seeing the importance of using reconciliation. So basically taking into account explicitly duplication, the losses, and also supertree approaches. And so usually what we ask ourselves is which model should we use? And for that you have to remember, do I have to take into account signal heterogeneity? Are the genes coming from different evolutionary histories? And are those genes of different sizes? So do we have to take into account paralogy explicitly? And is this a tall or a wide problem? So do we have a lot of samples or do I have too few samples? And before finishing, just shout out. So if you want to know more, there's this recent book. I thought this was an amazing work. I'm not directly involved in this. I have colleagues who are involved in this, and it's a very nice book in all aspects. So you see, it's a dream team of researchers, and there's no money involved, so there's no commercial publisher. You can just go there, download a very nice book for free. And yeah, I would like to finish by thanking you and also thanking my group. So these are the Binfies at the Quadram.
all right. Thank you so much, Leo.
Thank you. Oh, you're there. Okay.
Yeah, I'm there. I'm back. Yeah, I just exploded for a second. It's all right. Seems like everything went fine without me. So yeah, so that's good. On time. I think we got time for one quick question, but there are a couple which I'll leave in the Discord. The question is, in reconciliation, how do you account for the different rates of duplication, loss, and HTT in different parts of the tree?
So the methods that I mentioned here, they are just based on the cost. But there's so field dog and so for instance, field dog uses reconciliation in a way, and because it finds this most parsimonious reconciliation, but then it takes into account different rates in different genes. But yeah, the way that I describe it here, you don't take into account different rates. You just find the minimum cost one. So it's kind of a minimization. But you can incorporate into this model. So I don't know, you can, so for instance, my model here, it's not exactly a reconciliation, but some of the distances are the reconciliation distances. And then in this case, each one of the genes will have a different lambda. So this lambda here somehow takes into account the propensity of this particular gene to have duplications and to have losses. And SPR is a metric that is associated to horizontal gene transfer. It's not exactly that. But so if I would to use this method, these are the parameters that correspond to different rates. But as I said, there are different models like field dog, and I think in this book they have a chapter talking about that. But yeah, it's a very good point. Thanks.
Okay, excellent. Yep, so there's a lot more questions in the Discord in conference room 1 if you want to have a look at those, and people can continue the conversation there. Thank you so much, Leo. That's great.
Thank you, folks. Bye-bye.
And Rebecca,